I'd like to now introduce Dr. Will Hendricks. As a cancer scientist, Dr. Hendricks' vantage point in the human cancer precision medicine revolution of the past 20 years has shown him how the use of cancer genomics can save millions of lives. Motivated by the loss of multiple pets to cancer and his experience of the distress that comes with having very little information to help guide decisions on how best to help them, Will has been driven to bring precision medicine innovation to veterinary oncology, where millions of pets are diagnosed with cancer each year. Dr. Hendricks is the founder and chief scientific officer of Vidium Animal Health, as well as an assistant professor of integrated cancer genomics at the Translational Genomics Research Institute in Phoenix, Arizona. He completed doctoral and postdoctoral training at Johns Hopkins School of Medicine in Cellular and Molecular Medicine. In 2013, Dr. Hendricks established a laboratory at TGen to develop programs in canine and human cancer precision medicine alongside Dr. Jeffrey Trent. He has worked with a broad collaborative network to perform seminal genome landscaping studies in dog cancers and drive development of new treatments and diagnostics for dogs with cancer. He founded Vidium Animal Health in 2020 to accelerate the translation of genomic discoveries into the veterinary clinic. As always, please post your questions for Dr. Hendricks in the chat box during the presentation, and he will answer as many as possible during the live Q&A after his talk. Welcome, Dr. Will Hendricks.
So thanks to Dr. Boyko and the Embark team for putting together this important summit. I'm really excited to be able to join this group to talk to you all about the role of genomics in veterinary oncology at a time when this field is rapidly expanding. And this has huge implications for pet owners, breeders, and clinicians. I happen to work on the disease side of the canine health spectrum. I jump in after suspicion of cancer is already present, rather than in the disease risk prediction or wellness side of the spectrum, like Adam and others. But these two pieces of the genomic medicine puzzle are really intricately linked. And given the exponential growth in new data in canine genomics that we're seeing alongside a number of growing commercial genomic offerings for dogs, it's really more important than ever for our various communities to be exchanging ideas, sharing data, and finding ways to work together to leverage the remarkable advances of the past 30 years in genomics. Today, I'm going to spend about a third of this talk on the fundamentals of cancer genetics and genomics to set the stage for understanding how we use genomic information across the spectrum of screening, diagnosis, and treatment. I'll then talk about how human cancer medicine has been leveraging genomics, and I'll wrap up by reviewing what we're learning about canine cancer genomics and what we can do with this information. First, we all recognize that cancer is an increasing problem in our pet dogs. There are millions of cancer diagnoses every year, and many more that probably go undiagnosed. In fact, there's very likely a significantly higher incidence even than in human cancer. We also recognize that staying alert for and preventing cancer is really a daunting process that's eclipsed only by the subsequent challenge of actually navigating the landscape of that cancer diagnosis for a breeder, for a pet owner, and even for a veterinarian. We also recognize that we need more tools to understand cancer predisposition and risk, to support decision-making for cancer patients, and ultimately, we need more tools to improve outcomes. This is where genomics comes in. Cancer is fundamentally a genetic disease, and this means that we can use the rising tide of genetic information and genomic diagnostics to tackle all of these aspects in the care continuum. Genomics is immensely complex, but it's far from being out of reach. By the way, I'm probably going to be somewhat interchangeably using the terms genetics and genomics today, but often what I mean by both is the science of the study of genes and their role in disease, mutations in those genes that cause disease, and when I use the omics suffix, I'm talking about the complement of all the genes, the genome, in any individual, and how that full complement of genes and mutations contributes to disease. I'm also going to be using this landscape or map analogy quite a lot today. This is because it helps me and hopefully helps you visualize the complex genomic data that underlies cancer. Here, for example, I'm showing you two examples of how this complex but ultimately understandable these data can be. On the left, we're looking at an image of a full genomic complement of mutations that occur in a single canine oral melanoma. This is data from next generation sequencing of the DNA from this dog's tumor when compared to the DNA from his normal non-cancer cells. What I'm showing you is each of this dog's 38 chromosomes plotted around the outside of this ring. And then each of those blue triangles on those chromosomes, that's the location of an A, T, C, or G, a nucleotide in the DNA that's been mutated to a different A, T, C, or G, and is potentially playing a role in driving that cancer. Then in the middle of this plot, we've tracked areas where entire pieces of these chromosomes have extra copies or have no copies. And finally, these lines in the middle, those blue lines, they show where pieces of one chromosome has been broken and attached to a piece of a different chromosome in the cancer cells in this tumor. Look at how complex just this one genome is. But have no fear, because we can actually increasingly make sense of this and put it to use in the clinic. Part of how we do that is represented on the right. Here, this is data from sequencing the tumors of many dogs to find mutations in genes that are shared based on cancer type, on breed, on how aggressive the cancer may be. And we're then able to study these genes and mutations in the lab and in clinical studies to find out how we can use them to guide clinical management of individual patients. These studies also help show us how complex cancer is between patients, even with the same tumor type. Okay, so let's review the fundamentals. Cancer is a genetic disease, but that's just the foundation. Cancer is ultimately a disease of excess cellular growth. Its evolution gone awry from the perspective of the affected organism. Population of cells acquires a selective fitness advantage amongst all of its peers, and then it proceeds to take over and wreck the entire organism by growing and spreading out of control. Ultimately, that excess growth is driven by mutations in genes that control cell life and death. Under normal circumstances, these genes are just doing their regular job, growing when they're told, sending the right signals to their neighbors, behaving themselves. But when mutated, they can become always on, like a broken light switch or a gas pedal, triggering increased cell division, invasion, metastasis. I'm showing that process schematically here, in which a sheet of normal epithelial cells on the left, these could be the cells that line the inside of an esophagus or your colon, for example. One of these cells acquires a mutation in gene a cancer gene, that when mutated makes the cell behave aggressively. That mutant cell population then grows a bit more quickly than its neighbors. Sometimes other cellular machinery shuts down this mutation-induced excess growth, but other times additional mutations will arise in other cancer genes, and now this population of cells expands in clonal waves. As more mutations and more genes accumulate, a tumor forms, with cells all directly related to one another in the tumor, but increasingly different from the normal cells surrounding it. Eventually, this rogue population, this tumor, invades surrounding tissues, attracts the growth of new blood vessels because it's growing so fast it needs more help and more nutrients, and then cells can break off into the bloodstream and spread to distant sites. This is called metastasis. So I told you that cancer begins with mutations in genes that control cell life and death. Let's talk more about those genes. First of all, though, recall that we humans and dogs have 2 copies of every gene in our cells, 1 copy from mom and 1 from dad. This is important because sometimes a mutation in only one of these copies can drive cancer, and sometimes both copies have to be mutated to drive cancer. So we drop these genes into 2 categories based on the fact that they can occur in one or the other of these patterns. And we liken them to the cellular gas pedal and the brakes in a car that has 1 gas pedal and 2 brake lines. So bear with me on the analogy here. Some genes, the gas pedal genes, are oncogenes. Their normal function is a positive regulator for cell growth. As we grow and develop, they do their job of making more cells. If we mutate one of those, and we only have to mutate one, it can stick that gas pedal to the floor and accelerate cell growth. On the other hand, in the second example, we have tumor suppressor genes. These are the brake line genes. These genes normally are negative regulators of cell growth. They're the ones that keep the cells from growing out of control. If we mutate one of these and it stops working, we still have another working brake line. But if we cut both lines, then we've lost our ability to slow malignant cell growth. To bring it back to what all this molecular and genomic stuff means for an organism, mutations in oncogenes and tumor suppressor genes lead to excess cell growth, tumor formation, and sometimes then to the growth of malignant tumors that invade blood vessels, as this image on the right is showing for that primary cancer that's invading the bloodstream. And then they spread to distant organ sites. It's ultimately metastasis that most often kills cancer patients. Some of us now take this genetic understanding of cancer for granted, but it's not all that long ago that we actually realized that genetics is what is the driving force here, and that we've understood it this way. This is because cancer is a complex disease that has a lot of endogenous and exogenous environmental influences. Also, when I talk about cancer as a genetic disease, by the way, I don't mean that it's all inherited. Only a subset of cancers, about 10%, are heritable. The rest are sporadic, meaning that the mutations that cause them arise in initiating cells in the course of our lives, and they're not actually inherited from mom or dad. So for many years, cancer heritability research followed its own line of inquiry, while associations of cancer incidents with viruses, with hormones, with radiation, or with cigarette smoke or sunburns or other factors, they all followed their own lines of inquiry in parallel but separately. But what's ultimately been shown to knit all of these factors together is genetics. Every one of these associations has a direct link to genetic damage, to mutation. Aging is actually the greatest risk factor for cancer. This is because every time one of our cells divides, it has to copy all 6.4 billion letters of DNA. And the machinery that does that work is pretty accurate, but occasionally makes mistakes. Usually those mistakes don't have any consequences, but if one of those mutations occurs in a cancer gene at the right spot, that gene can then get hyperactivated and it can kick off that cancer process. This is actually the replication stress shown in this figure on the left. Other normal bodily processes can actually induce mutations, like inflammation or even regular metabolic waste products. We also discussed inheritance of mutations from mom or dad, that's shown in the middle there. And finally, mutations can arise in the course of our lifetime from outside sources like UV or radiation, cigarette smoke, and even some viruses like HPV that can insert themselves into our genomes and disrupt cancer genes, leading to mutations in those genes. So again, some of these mutations are somatic, meaning that they occur in only minor subsets of our somatic cells sporadically in the course of our lives. They're not inherited. And some of these mutations are germline, meaning that they were inherited by our parents' germ cells, and so they've been there from our very beginnings. They're present in all the cells in our bodies. They're inherited. It's important here to dig deeper into how we can acquire mutations through these two routes, because this understanding is going to be critical for how we think about using information on mutations for screening and or for management of active cancer cases. So somatic mutations are only in actual tumors, actually in the tumor, so they require analysis of tumor DNA itself for the management of those active cancer patients. Whereas germline mutations are present in all cells and can be detected in blood, saliva, or other tissues through screening tests. So the final step in our review of cancer genetics, thanks for sticking with me on this, is going to zero in on the relatively smaller subset of human cancers that are inherited, that arise from those germline mutations. It's important for us to recognize that not all mutations are created equal. There are some mutations that are so powerful that inheriting just one bad copy from mom or dad virtually guarantees that we'll develop cancer in our lifetime, and probably multiple cancers, and probably at a relatively young age at that. This is Mendelian inheritance after the famous geneticist Gregor Mendel. I'm showing you a pedigree here on the left in which dad, the top square, was the carrier of an autosomal dominant mutation in a cancer gene and died young of colorectal cancer. He was affected, his box is black. We then see that 2 out of his 4 children, in a classic pattern, were also affected, and then a daughter on the left who was a possible unaffected carrier with a white circle, whose child was also affected. Importantly, in these cases, we can often identify the responsible mutations to track their presence in offspring and to manage risk. These kinds of mutations tend to be very rare in human populations, otherwise we'd all have cancer at a young age and no one would be able to reproduce. These also tend to occur in tumor suppressor genes in one of the dual break lines in the cell. But this is the straightforward example of heritable cancer, a cancer syndrome in a family. This occurs in about 5% of human cases, familial cancer. We also see a lot more complex inheritance. Here, the pedigree is not so obvious. On the right, cancer is common enough that just because multiple family members are affected, in this case, we don't know if this is genetics, is it a shared environment and mutagenic exposure, is it the bad luck of being human? This is because a lot of mutations are less powerfully malignant, and it takes several of them, or takes several inherited mutations plus additional mutations from endogenous or exogenous sources, or from complex interactions between genes and environment to actually initiate cancer. But they do nonetheless increase risk. These mutations are actually much more common in the human population because they're not weeded out by natural selection. So why does any of this matter? It matters because this is fundamental to how we conduct research to identify these disease-causing mutations. And also it matters because it's fundamental to how you use genomic testing. Testing for known common inherited mutations or regions of a mutated genome for screening or general wellness often captures these known defined genomic positions. And increasingly, we're able to identify more of them as these positions are connected to disease. But there's also a huge array of possible rare mutations, rare variants like truncating mutations or disruptive mutations that break tumor suppressor genes that aren't captured because they're not common. And there are so many that we'd have to use more expensive technology to look at every single one of them. To me, this means that we might be underappreciating the amount of Mendelian disease out there in dogs, where pedigrees with lifetime outcomes data are not always followed, such that they point us towards these possible cancer syndromes. Now, most of the rest of my talk is going to focus not on screening or inheritance, but what we actually do with the information that comes from sequencing tumor DNA. But I wanted to leave this stake in the ground on an important intersection point for us somatic cancer genomicists like myself to talk further with the germline genomicists. So we've discussed how mutations in cancer genes can cause cancer, and we've discussed the sources of those mutations with some extra time spent on heritability. But how many genes are we talking about here? Well, there are about 20,000 genes in human and canine genomes, and based on comprehensive cataloging in a public database called Cosmic, of the more than 1.4 million human tumors that have had some kind of genetic analysis performed, we know that more than 700 genes have a role in cancer. That's quite a lot. So how does one go about profiling all of these genes to guide discovery and for diagnostic purposes? Hopefully this review of cancer genetics has gotten us prepared to dive into this enormous quantity of cancer genomic information out there and make some sense of it. A key to this is the technology backbone that's allowed us to get where we are today. The backbone of the genomics revolution is that of new technology that allows us to look at the billions of DNA letters in any individual or any tumor in the course of only 1 or 2 days. It allows us to do this in a massively parallel fashion, such that dozens to hundreds of these genomes are being sequenced at once. 30 years ago, as the Human Genome Project forged ahead into this brave new world, they were using technology that required warehouses full of pipetting robots. There's an image of one of those shown up here on the left, and capillary electrophoresis-based sequencers to amplify and sequence about 100 DNA letters at once. Now, if you stack enough of these together, you can eventually, 10 years later, and for billions of dollars, generate a full genome of 3.2 billion bases, as the Human Genome Project did. But through innovation in microfluidics, optical imaging, molecular biology, and chemistry, we can now collapse that warehouse onto a single slide that's loaded onto an instrument about the size of a washing machine, where those billions of DNA letters are sequenced all at once on a single chip. This massive disruption happened around 2007. You can see on the right how the cost of sequencing went down actually even more quickly than Moore's law of technology disruption. Around 2007, when this technology was introduced, this opened a fire hose of new genomic information that completely upended our understanding of many diseases, but particularly cancer. This technology is what took us from this idea of cancer, where we knew about a few dozen genes in the 90s and early 2000s that were associated with specific advanced tumor types, to this understanding of cancer, in which through sequencing of dozens, then hundreds, and eventually hundreds of thousands of human cancer genomes, we discovered that cancer was far more heterogeneous than most of us ever expected. Hundreds of genes mutated in varying patterns across tumor types and even within individual tumor types, and even within individual patients. Here I'm showing you the results of a sequencing study of the entire tumor genomes and matched normal genomes from a couple of thousand of human cancer patients across cancer types. Each of these columns is a tumor type for which 100 or more tumors were profiled. Each of these rows is a cancer gene. The color of the box indicates the proportion of patients' tumors that had a mutation in the gene shown in that row. The number is the total number of patients. So we look at this first box on the left, upper left, to see that this TP53 gene P53, a tumor suppressor that we know to actually be the most commonly mutated gene in human and canine cancer. This gene is mutated in nearly all breast cancer patients. We've got that dark blue box under breast adenocarcinoma. But it doesn't show up in the middle there in any pilocytic astrocytomas, a type of brain tumor. Much like that first plot, my first slide that looked at this kind of stuff in 3D, you can imagine peaks and valleys here if you looked at it in 3D, with the dark blue spots, the peaks, and the white being valleys. Look at this diversity across cancer types and look at the diversity within a single tumor type in a column. This genomic diversity means a couple of things. One, cancer is far more complex than we suspected. And even within a single tumor type that looks the same under the microscope and behaves in the same way clinically, we might have different mutations in 2 different patients. This has huge implications for drug discovery and development because it means we aren't actually likely to find magic bullets that work for all patients. But all hope isn't lost here because point 2 is that there are shared mutations across cancer patients. This genomic diversity also underscores the huge need for genomic diagnostics. Well, why do we care about these mutations at all? So hopefully I've helped you understand the genetic basis of cancer and what we're learning in human cancers. But why do we care so much about this, even if these mutations are linked to the initiation of cancer? Well, we've talked about how they can drive tumor growth, but let's now talk about how they can impact treatment itself. So one important example of a human oncology drug that I'm giving here is gefitinib. This was an early example of a genomic-informed therapy that we almost didn't have because its development started in the pre-genomic era. Gefitinib's trade name is Iressa. It was developed in the late 90s and conditionally approved by the FDA in 2003 as a third-line therapy after chemotherapy in locally advanced non-small cell lung cancer. It was developed to target a gene, the epidermal growth factor receptor, known as EGFR. This development was based on biologic principles, knowing this gene's operation was important in lung cancer, even though we didn't know anything about mutations that might occur in that gene at the time. But the approval was withdrawn 2 years later due to a lack of evidence that it extended life in a large follow-on clinical trial, even though during that trial there was a subset of some remarkable responders. So during the same time period, there were a few labs that were publishing reports using genetic techniques that showed that between 10 and 15% of non-small cell lung cancers actually had mutations in their EGFR genes. And those mutations were determinants for gefitinib response. In 2008, this was confirmed in a large follow-on study with over 12,000 participants that showed in a subgroup with mutations in EGFR, gefitinib as a first-line therapy resulted in significantly longer progression-free survival. Conversely, the patients who didn't have this mutation did better on traditional chemo. This was the first time a targeted single agent, a targeted monotherapy, demonstrated significantly longer progression-free survival than chemotherapy. And this slingshotted the world of targeted therapy forward. There have since been additional phase 3 trials confirming the superiority of this drug over standard chemo in the mutation setting, and lots of development in this field with even better drugs coming out in the past years as well. But the point here that this was only about 10 to 12 years ago that we really started to be able to separate these patient populations by mutations to look at the benefits of targeting those mutations. This was also the first big case of a drug looking for a companion diagnostic or a specific mutation to show where the drug's going to work. And it's not alone. There are similar stories for imatinib from the late 90s, double survival rates for leukemia patients bearing a particular translocation, the Philadelphia chromosome, cetuximab for colorectal cancer patients that bear mutations in EGFR but not in KRAS, et cetera, and on and on and on. So this is a big reason that we care so much about cancer gene mutation. Let's also not forget, as we've already discussed, that we care about mutations in cancer genes because when they occur in the germline, they confer cancer risk. Think of the example of BRCA, with which we're all probably familiar, in human breast and ovarian cancers. Again, I'll be focusing on the somatic side of life, but clearly this is an important part of the equation. So let's step back to the bigger picture to review how mutations have clinical utility and impact for cancer patients. They're not only valuable as companion diagnostics for targeted drugs, but they can often be used to determine or supplement a diagnostic workup. let's think again of that Philadelphia chromosome translocation I mentioned in CML and leukemia. Here it's shown through a fluorescent chromosome staining technique that highlights the BCR gene is green and the ABL gene is red. These two genes aren't supposed to be stuck together because when they get stuck together, they cause cancer. Here in these blue cell nuclei from a smear from a possible cancer patient, we see them stuck together, and so we're able to make a CML diagnosis. Mutations can also provide prognostic information. Here's data on mutations in the p53 gene, Kaplan-Meier survival curves that show that when we compare the mutation status of p53 in the graph on the right, and whether the wild type or the non-mutated gene in blue, how that compares to tumors bearing the mutant gene that's shown in red there, how long do those patients live? And that red line is dramatically lower than that blue line. So patients bearing the p53 mutation have much shorter, significantly shorter survival than those who don't bear the mutation. Compare that to some of the standard staging tools that are used, like tumor size on the left, and we see that these mutations can be equally as informative as other staging prognostic techniques. And again, finally, mutations can predict response to therapies. We've talked about gefitinib. Here I'm showing another example. Gefitinib was sort of a retrospectively resurrected mutation-based companion diagnostic. But in this case, I'm showing you an example of a targeted drug with a mutation-based companion biomarker for whom development was actually led by the discovery of the mutation. So in the early 2000s, we started to see through genetic and genomic techniques that mutations in a gene called BRAF were very common in many cancers, but particularly common in melanoma. There's a pathway diagram on the left that shows how if we target that mutant BRAF gene, we can shut off cell signaling and attack cancer. So this drug, vemurafenib, was developed to target the BRAF gene specifically because we knew BRAF was so commonly mutated in melanoma and many other cancers. In the middle, you see a remarkable, striking CT scan of a patient treated with this drug who had BRAF mutant melanoma. On the left, we see metastatic disease, and on the right, we see a lot of that disease resolved shortly after treatment. And on the far right, we see survival curves. At the time this drug was developed, melanoma was still treated with a cytotoxic chemotherapy. You can see patients treated with this drug had dramatically improved survival relative to the patients who were treated with the chemotherapy. So this paradigm is universal now in human cancer drug development, companion diagnostics, and the standard of care for cancer patients. In fact, there are now over 200 FDA-approved cancer drugs, and many of these bear mutation-based companion diagnostics, and this number continues to grow dramatically. Ultimately, this genomic paradigm has led the way in the progress the cancer medicine field has made in humans in reducing cancer deaths. This reduction is due to many factors across the continuum of care, from prevention and screening playing a big role here, but also significantly to development of new treatments over the past 20 to 30 years, and new approaches in surgery, other non-targeted drug development, radiation as well. But I'd really argue that genomics is the framework underpinning most of these advances over the past 30 years, because it's genomics that's guided how we have developed and deployed risk assessment tools, new screening and early detection tests, genomics that's guided new drug development, and the hundreds of targeted therapies that are prescribed based on the presence of companion mutation-based biomarkers. It's also genomics that's increasingly forming a component of the diagnostic workup of any human cancer patient. It's now common at most comprehensive cancer centers, academic medical centers, and even in community oncology centers for some type of mutation-based profiling to be offered for cancer patients. Dozens of companies have also risen up to develop and offer commercial cancer genomic tests to help manage human cancer patients and improve outcomes. Okay, so enough about human cancer. What does this paradigm shift mean for veterinary medicine and for our pets with cancer? First of all, let's go ahead and commit to the fact that cancer is also a genetic disease in dogs. Although we don't have as much data in dogs, we do have lots of points of data that support that this is true. Here I'm showing data from a study that looked at mutation patterns in a common tumor type, mast cell tumors in dogs, and looked at the differences in mutation patterns between low-grade mast cell tumors and high-grade mast cell tumors that are more aggressive. If we look at that top row, what we've done is we've plotted the different canine chromosomes along that bottom axis. And if we have extra copies of a gene, we've got a blue line sticking up. If we've lost copies of a gene, if a piece of DNA has been deleted, it's a red line sticking down. What we see is in those low-grade tumors, there are significantly fewer genetic events, genomic events. In those high-grade tumors, there are a lot more events. This shows us that we accumulate mutations that correlate with the aggressiveness of cancer, and this helps support the cancers of genetic dogs. Again, we have abundant other lines of evidence to support this, even beyond the basic first principles of heritability, evolution, cancer behavior that connect these two. One last foray into the germline side of life. We also know that cancer is a genetic disease in dogs because much more so than in human populations, we see clear linkages between particular breeds and cancer. I'm showing here data on the left from the recently released white paper from the nationwide vet analytics group, quantifies the risk across breeds based on cancer claim data. All of this makes sense from a genetic perspective in which risk is unique to and elevated in closed populations. As we increasingly understand the genes and mutations behind this risk, this can be a powerful tool to inform screening and breeding practices. So how much do we know about mutations in dog cancers? Recall that I showed you the 1.4 million human cancer cases that have been profiled. Well, the community at large has also profiled over 3,000 canine cancer genomes. So we have a lot more work to do to catch up to human, but we already know enough to understand quite a lot about the genomic underpinnings. Let's look at a specific example. Lymphoma is one of the best characterized canine tumor types genomically, particularly because it's so prevalent. And we've learned a lot from genomic studies of lymphoma that already hold clinical value. And we still have a lot to learn. Here, I've summarized 8 studies, and I'm showing you a plot somewhat like the human pan-cancer landscape plot we looked at. But here we've got one for just one cancer. So it's like taking one of those columns and sticking it up in another dimension. We're comparing B-cell lymphoma in the front to T-cell lymphoma in the back. What we can already see is that there are some shared peaks. We see that MYC gene on the far left, that shows up in both tumor types. And we see others, if you look further in, that TRAF3 gene, that shows up in B-cell lymphoma, doesn't show up in T-cell lymphoma. So there are shared peaks, there are unique peaks between these two subtypes of lymphoma. Some of these genes are involved in predisposition, some are involved in and can be used to support diagnosis. Some are even new and emerging drug targets. So this is the kind of information that we're getting out of genomic studies in canine cancer. So how many mutation-based biomarkers do we see overall? In the human space, we have over 400 biomarker genes, 700 genes that are involved in cancer. There are many clinical trials of new drugs matched to mutation-based targets. In the canine space, we don't yet have as much going on, but this is all starting to change. We already know that there are over 100 biomarker genes that have been identified in canine cancers based on peer-reviewed publications. We know there are more than 1,000 biomarker mutations in those genes. So the number of these is growing every day. These data, alongside technology innovation, have led to the development and commercial release of a growing number of canine cancer genomic diagnostics as well. Again, these span the spectrum of care, from screening tools that help us with cancer prevention, risk prediction, and even new assays for early detection of cancer, into the diagnostic setting, which is where my focus is for profiling active cancer patients and helping with treatment planning, and then even to the disease monitoring stage. As we're able to do more for cancer patients and improve outcomes for those patients, it's going to be increasingly important to watch for development of resistance mutations or our ability to look for minimal residual disease. In each of these settings, with the exception of the monitoring setting, there are at least several commercial assays that have been released in the past 5 or so years, including those that are used for wellness, for prevention, and as I mentioned before, even early detection of cancers based on liquid biopsy. And then in the diagnostic setting, there's a growing universe of tools being used to help guide treatment planning for canine cancer patients as well. What does this kind of testing look like in practice? So I know that you're all familiar with Embark and likely also with genetic screening tests in general, but how does genomics work in the setting of an active cancer case? Here's an overview of how Vidium's flagship product, a tumor genomic profiling test called Searchlight DNA, operates. A sample of the tumor, we start in the bottom or the top middle. A sample of a patient's tumor is sent to a pathology lab, or in addition to being sent to a pathology lab, it's a sample like an aspirate is sent to a clinical genomics laboratory in parallel. DNA is extracted from that tumor. The DNA is sequenced. Often it's in a targeted panel of genes. That data is then undergoes a comprehensive bioinformatic analysis to find mutations that might have some clinical value for that patient. A report is generated and delivered back to the veterinarian and the clinician as well. And what these reports do is we find mutations in this patient's tumor, and then we map them to a database of known peer-reviewed biomarkers and biomarker information on those mutations. This can point to, okay, if we found mutation X, is there a role for that mutation supporting a diagnosis? Is there a role for that mutation in providing prognostic information? And might that mutation also be linked to a new treatment option that can be accessed through a compounding pharmacy? So let me wrap up by showing you two examples here of this kind of tumor mutation profiling in action. In the first case, we have an osteosarcoma where the genomic profile of this osteosarcoma biopsy showed a broad number of mutations, some that we expected to see that we know commonly occur in osteosarcomas based on what's in the literature and the work that we have done, like mutations in genes like P53 or SETD2, et cetera. We also found mutations that have prognostic information, either directly from the canine literature, like that P53 link with breast cancer outcomes I showed you. There's data in osteosarcoma as well, and even mutations that are linked with treatment options. Now, could we have guessed that any of these mutations would actually occur in this tumor? We could have, and with varying degrees of success, but this unique array of mutations that occurred in this particular patient's tumor required this level of deep genomic analysis in order to bring all the information to the table to make the best decisions. Let me show you one more example here. From a squamous cell carcinoma, and this is actually an example of how genomics can provide diagnostic guidance. In this case, there was a concerning nodule found on this dog's head in June of 2021. After histopathology, it was suspect for, but not definitively, squamous cell carcinoma, and a cyst was still in the differential, even though there were signs that it was invading bone. The veterinary team started the dog on Pladia before running the Searchlight DNA assay, just based on literature that suggests that Pladia's target, the KIT gene, is activated in this tumor type. Searchlight profiling showed the presence of significant mutations that are associated with malignant cancer to help support that malignant cancer diagnosis. They also identified other mutations associated with a poor outcome and potential treatment options linking the mutations in this tumor to targeted therapies available through compounding pharma. So this, again, is an example where tumor mutation profiling can provide diagnostic guidance and also therapeutic options for it. So hopefully I've given you a sense of how at least one piece of the equation can be used in practice. And while much of my talk is focused on what we can do with genomics for these active cases, it's important to leave you here one last time with the recognition that there's interdependence between germline cancer genetics, screening and testing, and the somatic side of life and diagnosis and monitoring of active cancer cases. We have a lot left to learn and a lot that we can do together where these points meet. So I hope I've left you with an understanding of how genomics is broadly changing our understanding of human and canine cancers. I hope I've showed you that clinical genomic testing in canine medicine has arrived with abundance of new tools available to veterinarians, breeders, and pet owners, and that genomics has incredible utility for the management of canine. So thanks very much for your attention, and I look forward to discussion.
Hello, everyone, and welcome to the live Q&A portion of this talk on cancer genome landscapes, how DNA is changing the way we treat cancer. Joined by Dr. Will Hendricks. Thank you very much for that informative presentation.
Yeah, thanks, John. Thanks so much, everyone, for watching, and it's really been a pleasure to be a part of this really great meeting.
So to start off, we have a few questions that have come in so far, and just as a reminder to everyone, please feel free to continue posting questions. We will try our best to get to as many of them as we can. To start, as I highlighted in my introduction that I read for you, you've had a pretty interesting, I think, career and path. Would you mind just kicking us off by sharing how you kind of got into the field you're in and why you're so passionate about it?
Sure, John. My professional path has definitely been a winding one, I think, like probably many of us here, but to try to make a long, winding story short, somewhere at the intersection of a love of genetics, probably having read too many X-Men comic books and science fiction as a kid, and still probably reading too much of that stuff, and really a desire to tackle big scientific and medical problems whose solutions actually have real-world translational impact. I think through all these lenses is where I ended up in this field of cancer genomic medicine, where I've worked for the past 20 or so years. I think, like most of the rest of us, I've lost friends, family members, and pets to cancer, and so I'm intimately familiar with that fear and frustration and feeling of helplessness that comes with the limited spectrum of options we have to even understand the cancers in our pets, let alone to treat them. At the same time, I've been fortunate to have gotten to work with some of the leaders in the field of human cancer genomic medicine, and to have had this front row seat to the revolution that's been going on over the past 20 years as we map the human genome, as genomics then really started to drive development of new drugs and diagnostics. And these advances have all been central to that 30% reduction in cancer death rate that I was talking about in my talk that's happened over the past 20 years. So I've had this sort of discordant experience where I've gotten to see human oncology research and medicine grow so rapidly on this foundation of genomics, while we're still sometimes arguing about whether cancer is even a genetic disease in dogs in certain settings in veterinary medicine. So ultimately, I've had those two different perspectives, and at the same time, again, like I think all of us in this field, I'm motivated by the fact that I've got an 11-year-old rescue, an Amstaff mix named Jake, who's this giant doofus of a dog he spends his nights moving from sleeping in each of my 4 kids' beds to keep him safe. And he's one of those lumpy dogs. And, I worry that any day now, one of those lumps is going to turn malignant. So I want to be able to have as many options on the table as possible for Jake as would be on the table for a human patient, so that he can have a long and happy life.
Yeah, it's highly motivating, and as you said, I think a lot of us can relate to this topic. And I saw, to your point on kind of the revolution that's happened, I saw one of your slides about the number of FDA cancer drug approvals by year has really been, accelerating the last few years. So getting into the audience Q&A, the first question I see here is there any progress in the Belgian Shepherd gastric cancer research, and what can breed clubs do to help?
Yeah, so it's a great question. So I don't work specifically in the setting of the Belgian Shepherd gastric cancer research. I believe Dr. Evans mentioned that she's working on that side of things, I think, from trying to discover genetic predisposing factors in that setting. What I can say about gastric cancer is that we certainly know a lot about it in the human space. We know a lot about it genomically, and it's actually a fairly complex disease, or even a spectrum of diseases, because once we start looking through that genomic lens, we see these different baskets, again, like we were talking about in the talk, and there are these certain major subtypes that have characteristic mutations in them. So we understand a fair amount about the mutations that occur in human gastric cancer, and there's been some work done to look at mutations in canine gastric cancer. Again, not talking about like from Dr. Evans' work, where she's looking for the genotype of the dog and what mutations might predispose. what we're interested in is what are the mutations that occur in the tumor itself. We've seen a number of gastric cancer cases through our clinical genomic assay in Vidium Searchlight DNA. And we do see a lot of similarity between canine gastric cancer and human gastric cancer in terms of shared mutations. And again, what that kind of helps us do is it opens up the options for using those mutations to better understand, diagnose, prognose, and even point to possible new treatments and generate new hypotheses. So that's where some of my perspective on gastric cancer comes into play. I think to also answer the other part of the question about what can breed clubs do on the somatic cancer side of life, what can breed clubs do to help advance research and understanding of the mutations that occur in the tumors themselves. So I think first I would say, of course, as you all already are involved in and know about, participation in citizen science projects is hugely helpful for advancing research. Again, many of those have been described over the past 2 days. Using Embark's products to genotype your dogs, using generally genotyping, you can also participate in citizen science that way. Similarly, in the case of active cancer cases, utilizing genomic diagnostics can also facilitate contributions to research. So you may have genotyped your dog when they were well, but I think critically important, what's really most important here is also bringing to bear genotyping and genomic diagnostics for the tumors themselves for those active cancer cases. And what I would say to breeders and breed clubs is one of the great things that you all are great at is education. At Vidium, we have a lot of educational material around what is cancer genomic diagnostics for those active cases at vidiumah.com. So would encourage you to explore the resources that we have available. We'd also love to chat more with you about what is this new universe of cancer genomic diagnostics and how can you put it to use. Really, ultimately, what we hope and want to see is that pet owners and breeders whose dogs have developed cancer are bringing genomic diagnostics for those tumors to bear, both to advance research, but also help with manage the individual patient's cancer.
Yeah, we were speaking kind of backstage before coming out for the questions here a little bit about this topic. And, as you mentioned, I think breeders and breed clubs both are really tuned into education and specifically looking for what are the best practices that they can apply today to contribute, and make an impact on this topic. And it sounds like one of the things you're saying that I think we chatted about before as well is there's a best practice that can be done today that wasn't really available 5 or 10 years ago, or at least not readily so, which is specifically when a dog is diagnosed with cancer, making sure the tumor itself is genetically tested. Is that right? Is that like the big kind of change, if you will?
Yeah, absolutely. I think you hit the nail on the head there. It's also ensuring that the tumor is genetically tested because that information can help the veterinary team diagnose, prognose, and identify, expand the possible treatment options.
That's great. And, as we know, breeders, not only with their own kind of direct dogs, but their network, between all of the puppies and new families that they go to, breeders can really help kind of share this information along with veterinary clinics and others. So next question here, are the cancer genetic analyses currently available better suited to some types of cancer than others? For example, is it as likely to produce actionable information in lymphoma as mammary cancer?
Yeah, that's a great question. So I think there's two sides to the answer from my perspective. One side is we know a variable amount of information about different cancer types genomically. We actually know lymphoma is one of the cancer types in dogs about which we know the most, and there's still a lot to learn. Mammary cancer, there's actually been a lot of information emerging on mammary over the past couple of years in dogs as well. So there's a variable amount of information, and there is a different spectrum of mutations in each cancer type, and each of those mutations has a different spectrum of actionable information. So let me give a couple of examples. Lymphoma is an area where there's a fairly well-established standard of care, and where we tend to see fewer mutations that could have associations with novel treatment options. And at the same time, for patients who have relapsed on the standard of care, there might be a broader sort of horizon of utility for any of those, alternative therapeutic biomarker associations. But lymphoma is an example where we also often see diagnostic utility for genomics. There, as I mapped out in the talk, there's a set of mutations that are highly specific for lymphoma. So if lymphoma, mast cell tumor, other round cell tumors are kind of in the differential diagnosis and it's not clear what we're looking at, genomics can actually help point specifically in one direction, and then making that diagnostic call will then guide the veterinary team down a different treatment path. So there's part of the point here is there are variable amounts of information, but the second part of the point is that this idea that genomic diversity underscores the need for genomic diagnostics. So the possibility that you might see mutation X in 10% of a certain tumor type that could point you in a certain direction is part of what motivates the need to do this. Since cancer is so genomically diverse, even within tumor type, we have this opportunity. If we do find these certain sets of mutations, it gives us very valuable information. So it's that the possibility of the highly valuable actionable information is part of the motivation for doing this individualized genomic approach.
Interesting. So there's a comment in that I think relates a little bit to another question I was going to ask you. So we could start with this. It basically references there's a dog, a border collie and Catahoula mix that has had cancer twice. The first time was spindle cell tumor or mast and spindle cell tumor. That was addressed via surgery, and then another type of cancer appeared later, aggressive cyst cell sarcoma. So I don't know if you can speak to that specifically, but what it makes me think of is what have we learned and what is still to go about cancer reoccurring? You have one type that may or may not be addressed, and then another type comes on. I know that's not terribly uncommon, both in dogs and people. So can you speak to that topic?
Yeah, sure. So I think that there's still clearly a lot for us to. I think I lost you guys for a second. Can you hear me?
I hear you. Yeah, I think you're good.
Sorry about that, John. Not sure what happened. I think there's still a lot for us to learn about predisposition to cancer. And we've heard from a lot of great speakers who are really advancing this, and the Embark team of scientists is advancing this field too. What are those genetic loci? What are those specific mutations that are predisposing our dogs to cancer? And some of those mutations, like I was talking about in my talk, have a really huge, they dramatically increase the risk of cancer at a young age, and even multiple cancer types. Some of them may be even in that familial category. I think we still poorly understand what that familial category might look like. So there's still more to learn there. And I'll tell you from the data that we've seen. So we do tumor profiling. We sequence the DNA in tumors. But I'll tell you that based on what we've already seen today, at least 5 to 10% of our cases, we find mutations that really look like a smoking gun for being what we call a pathogenic germline variant that would be a high risk variant. So I think there's a lot more of that out there than has been appreciated, and more work to do to map all of that out. So, and particularly in the setting of when we have our pets or patients and they have multiple cancers or cancer at a young age, there's likely a genetic component to that in the predisposing setting.
Got it. So as a final question, we're about out of time, and I want to ask you kind of like a closing wrap-up question that I think we could probably spend all day talking about. But if you could comment on it, maybe just in 30 seconds or less. As you look ahead the next couple of years, what are you most excited about? How do you see genomics changing the way we care for cancer? And, any other innovations or changes that you see on the horizon?
Sure, John. Yeah, I think there's an exciting wave of new diagnostics, broadened access to drugs and information about those drugs, and the genomic data we're generating. I think that we have the tools, data, and resources today to begin to bring this low-hanging fruit from human oncology innovation. Again, biology, drugs, data, diagnostics, rapidly into vet oncology, and to start to dramatically improve quality of life and survival for our pets with cancer. I think in 5 years, I'd want to see a thriving genomics diagnostics industry just across the spectrum, from risk prediction and screening to this tumor profiling setting, and a growing wealth of information on and availability of these repurposed human oncology drugs that we already know work in the setting of particular mutations in human cancer types. I hope to see a growing pet cancer drug development industry that's also incorporating genomic discovery and biomarkers into that process. And ultimately, as a field, I think I'd challenge us through these innovations to reduce the cancer death rate in dogs by at least 30% in 5 years, as it's been done over the past 20 in humans.
Amazing, amazing. Well, I certainly hope that will occur and look forward to that as well. Overall, thank you again very much for your participation here. It's been really informative, and for everyone viewing, you can learn more about Dr. Hendricks through the profile on this conference site and via your company, Vidium. So thank you again very much.
Thanks, John. Thanks, everybody.
All right. And just as a reminder for all of our attendees, the next session has already begun. It's by Dr. Heather Hewson on Alaskan sled dogs, the genetics and selection of elite canine athletes. So you can join that session by going back over to the auditorium and switching over to that presentation. So thank you again for joining us here today.